So as everyone in this audience knows, that acinar-deductal metaplasia takes place in normal PDA progression. And during that time, there is progressive loss of proteins from the basic helix-loop helix transcription factor family, notably PTF1A, which is required for pancreas development, and in the adult pancreas, directly activates the acinar enzyme genes, and MIST1, which is an acinar maturation factor that regulates cell-cell contacts and the secretory pathways. So the way that basic helix-loop helix proteins activate EBOX sites in the DNA is they're obligate dimers. And they are regulated not only transcriptionally, as we see in PDA development, but at the functional level by the family of ID proteins, ID1 through 4, which lack DNA binding domains and sequester the VHLH proteins in inactive dimers. We and others found years ago that ID3 is particularly highly expressed in human PDA as well as in the Panin lesions in the KC mouse. We went on to look at models of pancreatitis and saw that ID3 is evident within 3 days of the duct ligation model of pancreatitis. So immediately when the cells become Ki67 positive and you start to see the ductal hyperplasia, ID3 is very highly expressed. And we looked at other models of pancreatitis as well. And this suggested a very early role for ID3 in pancreatic disease pathogenesis. We actually went on then to ask whether ID3 might be sufficient to induce cell cycle entry in absolutely quiescent populations of pancreatic exocrine cells. And this is a study we did in collaboration with Andy Lowy and showed that in the CK19 positive cells, none of which are pink for Ki67 here, we now, where the white arrows are, have essentially all of the cells that expressed the ID3 now entered the cell cycle. And some, as you can see in the graph and the images below, also express phosphohistone H3, showing that they're progressing through the cycle. So we did numerous studies with ID3 knockdown, and we also examined if ID3 was acting in the method we suspected by downregulating activity of VHLH proteins. Then we expected that if we restored VHLH activity by expressing one of those proteins, it would have an equivalent effect to ID3. And for the rest of the studies I'm going to show you, we used an inducible system so that we could activate the VHLH protein E47 upon demand with tamoxifen. Anyway, in either case, ID3 knockdown or induction of E47 induced immediate growth arrest in the PDA lines that we looked at, and this was primarily G1 arrest. What was remarkable to us is that the cells reverted toward their acinar origin. And these are cells that, for the most part, did not have PTF1A or MIST1 expression when we began the studies. And what you can see here is massive induction of trypsin, elastase, carboxypeptidase, and other enzymes. And here is trypsin expression in one of those cells. And in fact, just recently, a new assistant professor at UCSD looked at trypsin activity for us and showed that the trypsin is fully active in the cells. Another thing that was remarkable to us, and this was done in collaboration with Steve Konechny at Purdue, is that E47 induced feed-forward activation of the maturation VHLH MIST1. And here's MIST1 protein. What I'm not showing you, but is published, are all the downstream factors of MIST1 that were also induced. So we wanted to know if the same would happen in vivo, and what we used here were PANC1 cells that had not been induced, did not have inducible E47, or those that did, and all cells were treated with tamoxifen so that the PANC1 would have that control. And what I want to point out is that E47 was, had a profound effect on tumor growth. And moreover, in the controlled tumors, there is no trypsin expression that can be seen in those tumors. And they're pretty homogeneous in the Ki67 expression as well. We had cells that escape our E47, even when we sort for the cells that have the E47, there are always some contaminants. Those cells grew. But where the cells had E47, very few are Ki67 positive, and they uniformly express trypsin. So the next step was to move to PDX-derived lines. And this is more recent work that we've been doing with Andy Lowy. I tend to tell people in San Diego, all roads eventually lead to Andy if you're working in pancreatic cancer. And so here, these are two recently derived lines from Andy's lab. 779 is from a primary tumor. And in collaboration with Brian James and Garth at Sanford Burnham, and work done by Kathleen Scully in my lab, we've now shown that in 779, there's an insertion at amino acid 209 in P53, and this actually leads to an early stop. And we see no P53 protein at all. You know, most of the lines have the gain of function P53 mutations. Here's the P53. And then in terms of otherwise, the KRAS is similar. And then in the 1334 line, this is derived from a liver met. And what's unusual there, it's the only line that has protein expression for P16. And a more in-depth look at the exome sequencing for these new lines shows a myriad of other high-confidence mutations. And really the point I want to make about that is very few of these are in common with each other. And when you look at what's been published about the lines that are available from ATCC, BXPC3, PANC1, none of those are in common with those lines either. So they're quite diverse after the big four. Nevertheless, all 5 lines, including the metastasis line, react to E47 with immediate growth arrest. And we don't see evidence of regrowth. When we performed RNA-seq, again, this is 36 samples, 3 without E47 induced, 3 with E47 induced in all these lines. Actually, this group is 30. And the main category that's changed is cell cycle genes. One of the genes that is profoundly upregulated in all of the lines is P21. So this is the P21 in BXPC3 to begin with, and this is upon E47 expression, and so on. And we've shown that P21 is required for the growth arrest. Now, we also noticed significant changes in the cytoskeleton of the cells in response to E47. In particular, if you look at PANC1, it has that classical look of senescent cells, the large, flat cells. But some of the lines begin to die eventually in response to E47. And our thinking now, or our thinking upon seeing this, is that maybe all of the lines were initiating a senescence program, but they may not all succeed in finishing it. So I want to show you the SA beta gal, which is not a completely clear. The pink one that I mentioned, the huge flat cells, this looks like traditionally what we would expect. The cell lines that don't have the inducible E47 in them have very little SA beta gal to begin with. And we think that we're seeing some SA beta gal due to the slight leakiness that even in the lines where we haven't induced the E47, if the vector's there, even a small amount of leakiness may be causing some of these issues. Nevertheless, when we induce E47 activity, the SA beta gal increases, but we wanted to look at other markers of senescence. And what we're showing here is lamin B1 and Sen-PA that are senescence markers that have been identified by Campisi and others are quite downregulated by E47. P27 is, interestingly, did not change much at the RNA level in the RNA-seq data, but we noted that the ubiquitin ligase that's responsible for degrading it was severely downregulated by E47 and thought that the P27 protein, therefore, might be upregulated, and that's certainly the case. So there's and what's not here are SASPs that are upregulated by E47. So again, the thinking right now is the cells enter a senescence program, and they may stop in that program at various points. So the P21 and some of these other markers we were seeing suggested to us that maybe since P21 regulates CDK4/6, that phosphorylates RB to inactivate it, that RB might now be dephosphorylated and reactivated as the main tumor suppressor that remains in these cells without a mutation. And this is indeed what we see. We see less phosphorylation at the 807, 811, less at 608. And I'll show you total RB in another blot. But although it's a little difficult to see in the 779, we have shown it's definitely there, and it's dephosphorylated. And one of the things that we notice is that actually, in addition to whichever phosphorylation site we're looking at, that E47 then results in multiple dephosphorylations that change the migration of RB. And we have shown in a number of the lines RB is required for the growth arrest. And so as you might expect, these are E2F target genes that are direct targets where E2F binds to their promoters. And they're all downregulated by E47. So the other thing that was very important to us to investigate in the RNA-seq was this issue of Acinar reprogramming that we had seen in lines that have basically been in culture for about 30 years. Would we see them in the PDX lines from Andy's lab? And what we show here, these are, this is an unbiased list that we started with of genes that are downregulated 16-fold or more in pancreatic cancer relative to normal adjacent tissue. And not surprisingly, many of these are the genes associated with differentiation in those cells as they undergo ADM and lose Acinar characteristics. And what you can see here is they uniformly upregulate chymotrypsin and carboxypeptidases and elastase. And although this scale ends at 16-fold, some of these are in the hundreds-fold in the RNA-seq data. So that brings us to how will any of this help a patient? And something that we started was to develop a screening platform, which is now undergoing multiple iterations. But the first one was relatively simple in that we used 4 multimerized e-boxes, but not just any e-box, the ones that are specific for the subclass of BHLH proteins that includes E47. That's a very small subclass. And these are not the preferred E boxes for other notorious BHLH proteins like CMYC, or even for SREBP, because there are multiple families. And what we did is hook this reporter up to luciferase. And what I want to point out is deconvoluting what happens if we get increased BHLH activity or EBOX activity could wind up being a long process from an assay like this, because we might get increased BHLH activity by upregulating E47 expression, by downregulating an ID protein, by binding of a protein that phosphorylates IDs, and then that leads to a reduction in dimerization with E47. So the mechanism, we were agnostic to the mechanism. We just were looking for increased activity of this subclass of BHLH. And we ran a screen of known drugs, 5,700 from a total of 5 libraries. And here, if we're very stringent, these would be the hits above 6 standard deviations from the mean. What we then did is repeated the screen in triplicate for all the hits above 3 standard deviations from the mean. And what I want to point out here is that the top hit was a statin, and other statins were also hits in the screen, and that this suggests that it's a conserved mechanism in the chemotype. So we looked at the effects of statins on growth, and actually there's a fair amount published with statins, and looked at many of the genes we had shown that E47 regulates, and they're all uniformly downregulated. P21, again, is profoundly increased. And in terms of Acinar differentiation, though, this is where the data diverge. We do see some induction of trypsin in some of the lines, or elastase, but this is logs lower than what we see E47 can do. We do get some induction of MIST, again, that feed-forward activation of other BHLH proteins. But clearly, we either don't have as much BHLH activity, or we don't have the correct constitution of the BHLH proteins. Whatever the statins are inducing, we are getting RB dephosphorylation as described for E47, and a significant downregulation of E2F target genes, also signifying that RB is regulated. So to summarize, with 2 minutes left, we've shown that ID3 seems to play an early role in the pathogenesis and is sufficient to induce cell cycle in human pancreatic exocrine cells. Although most of our work is with E47, there may be others in this family that then regulate G0, G1 arrest, in part through P21. One of the players I didn't have time to discuss is TP53INP1, which we've also shown is required for growth arrest, and we haven't really worked on the mechanism of that yet. RB gets activated, and these top cell cycle activators are downregulated, and that also includes all the E2F target genes. On the other side, we see remarkable Acinar differentiation, which includes upregulation of most of the pancreatic Acinar enzymes, and feed-forward activation of MIST1 and all of its target genes. So the picture that we're left with is that BHLH signaling is the final common denominator between growth and differentiation in the cancer cell. And so where to go from here? Well, as I said, there's quite a bit published on statins in PDA, and although it's somewhat controversial, I think everybody would agree that it's clear that statins alone are not going to be sufficient to cure pancreatic cancer. But what we'd like to be doing next is taking all of the other hits that are from distinct chemotypes and testing them alone and in combination with statins to look for synergy in inducing the correct BHLH signaling that we at least know now that the cells have the plasticity that they can revert. And NCATS, a division of NIH, their screening program, they've chosen this screen now to look at 300,000 compounds, unique compounds, and that will be very exciting to follow up on. With Jim McCurrow in the pharmacology department here at UCSD, we're working on more complex cytometric screens since we know P21 is upregulated and we can see it at the protein level and that it's required. And we know that in some of the cells, the Acinar enzymes are reduced at a level that we can see well with immunostaining. And finally, after about 7 months of breeding and a lot of advice from Andy, we are about to have the E47 knockouts or ID3 knockouts on the KPC background. And this is where we'll really get to answer questions that we haven't addressed at all, including, well, if ID3 is knocked out in the stroma, is that still a good thing? We know in the epithelial cells it's pretty clear that it would be. And then, since our first screen was a repurposing library screen, then any of the combinations that we find do act in synergy. We have KPC mice to quickly move that along. So thank you very much.